If you wanted to make the most unique wine, something really special, well, firstly, you find a verdant, fertile country with young, untainted soils in a special location, so special that it has extraordinarily unblemished sunlight, pure, fresh, clear water, and just the right amount of hot and cold temperatures to grow fine, delicious grapes. Next, you make the wine. To make a unique wine, you need special people. People with a strong culture and connection to the land and a passion for sustainability. You need people with old world knowledge who are also innovative, dynamic, and unhampered by tradition. And they'd be curious and bold. They wouldn't stop making one variety of wine. Oh no, they'd be up for making any variety of wine. Pinot, Sauvignon, Chardonnay, and more. So who are these people and where does this wine come from? Well, some places have unblemished sunlight, fine soil, or beautiful water. Others have innovation, passion for the craft, or care for the land. But only one place has the purity of it all. New Zealand. It's altogether unique.
Hello, welcome to this session, which is part of New Zealand Wine Week. And the title of the session is Expanding the Scope of Sauvignon Blanc, Vine Diversity in New Zealand. And we have two really interesting panelists to talk on this topic. First of all, we have Daryl Lissamore, who is the lead of Grapevine Improvement Team at the Bragato Research Institute, and also Emma Maris, who is winemaker at Morisco and has around 800 hectares of Sauvignon Blanc under her control. So is well placed to talk about the importance of Sauvignon Blanc and also the vine material, which is a really interesting issue because, Daryl, if I'm correct, most of New Zealand's Sauvignon Blanc is just one clone, the UCD1 clone. Is that correct? Yeah?
Yes, it is. Yeah, we've been fortunate to find that this one genetic strain or variety just performs so well here, and that has meant that its uptake has been quite incredible. So the majority of what's planted is that one clone.
But obviously, Sauvignon Blanc is so important to New Zealand that obviously I remember a few years ago there was a lot of research on winemaking and how to work with Sauvignon Blanc, the science behind making Sauvignon Blanc and what it is that makes New Zealand Sauvignon Blanc so distinctive. So New Zealand's always been focusing on research, looking at how to kind of maximize that position as the number one producer of Sauvignon Blanc globally. So it's really interesting that the attention to be shifting to the vineyard now. Can you tell us a little bit about this project that you're involved with?
Sure. Well, as you say, there's been a lot of investment in the wine itself and the fruit and what produces the expression of these great characteristics. But the interesting thing in plant physiology is, and in genetics, there's always these two aspects of what produces any one trait. It's both the genetics of the organism and the environment that it's in. And we obviously manage the environment a lot. We grow vines in a special row orientation. We irrigate them. We do all we can to get the best out of them by managing that environment. We choose where in the country to grow them and which countries indeed. But we haven't actually done much with the genetics. The genetics of something like Sauvignon Blanc is hundreds of years old, and all that we've done is made these selections based on small trait differences that perform slightly better or worse in different places. And so, as you say, the one variant of Sauvignon Blanc that we've picked has done very well, but has been propagated en masse across the country. And that means in order to find any improvements or to make any adaptations, if we want to grow vines with less input or higher yield or look at new flavors and aromas, we need some diversity within the variety from which to select. So this project is really about supplying that diversity. It's been shown that it's really hard to bring in that diversity, not only because we have biosecurity challenges, but also because, as I say, the plants perform differently in different environments. We can't just take a grape that does amazingly well in France and expect it to do the same thing here. So we need a resource of diversity that we can select here in situ in New Zealand, in Marlborough, in the place we're going to grow it, and then pick the the better performing vines from those.
So, Emma, from the perspective of a winemaker, what's your need for new material? What is it about vine, Sauvignon Blanc diversity that would get you excited as a winemaker?
Yeah, so I think to your point earlier, Jamie, back to the mid to late 1970s, when my grandfather was involved in planting those first Sauvignon Blanc vines in Marlborough, not a lot has changed in terms of that vine material that we're putting into the ground, but we have pushed winemaking technique, we've pushed viticultural management technique. So I think, and we've really got amazing results out of that. We've got the Sauvignon Blanc that is known around the world as Marlborough Sauvignon Blanc. But what's really exciting is looking to the future, looking to future generations. And we are experiencing a changing climate. We are also in Marlborough really now drilling down on the many sub-regions and microclimates that we have. We are looking at ways that we can have less inputs in the vineyard and have manage our water more carefully, our natural resources, and pests and diseases as well. So I think what's really exciting is having a plethora of material now that's going to be available to us that we can start to really drill down on and dial in on what we're using and make really quite strategic choices about that vine material going into the ground that then supports, that's the fundamental starting point that then supports the process through the production of wine.
Yeah. So Daryl, plant breeding, traditionally plant breeding techniques, you'd make crosses and then you'd select the most interesting crosses, and you'd end up with a genetic diversity through breeding, through sex. But obviously with grapevines, we like to have certain varieties, and once you start crossing grapevines, you've got new varieties. So even if you were to cross Sauvignon Blanc with Sauvignon Blanc, you'd end up with lots of different varieties. And if you were to take the parents of Sauvignon Blanc and cross them, you wouldn't end up with Sauvignon Blanc again.
Exactly right.
This creates quite a challenge, presumably, for anybody working in plant breeding, because the only way to introduce new traits in a targeted way would be through genetic modification, because that's the only way you'd be able to keep the variety. Obviously, you can't do that. So how are you getting this variation, and how does it work in terms of mutations then producing new vines? Because obviously mutations are happening all the time in the vineyard, but presumably most of them are invisible or deleterious. To get something that really produces something quite interesting, it must be quite a rare event.
Yeah, that's a really good point. And plant breeding has obviously been hugely beneficial. If we look at the diversity of apples and ornamental plants and things that you see these days, that's because people have mixed and matched traits from parental plants. And you can do that in grapevine. A lot of people are doing that in grapevine, taking wild relatives to try and bring in disease resistance. But as you say, then it's not the same variety, and even siblings are not genetically identical. So you have to come up with a new name, and that name is a really strong signal of quality. People are buying wine based on the varietal identities they see on the bottle. But there is another source of genetic diversity, and as you say, that's the fact that vines do spontaneously mutate. Their DNA changes when they're sitting in when they're growing, when they're sitting in the vineyard, and that's because every time a cell divides and becomes two cells, it has to copy all of the DNA, and that happens an awful lot of times in the growth of a vine. And the other really key aspect of that is that environmental stress affects the rate of that genetic change. So the plants are quite genetically stable if you manage them in an optimum environment, but when they're in an adverse environment, an environmental shock causes that rate to massively increase. And that makes sense. Plants can't run away from an adverse environment, and like animals can. They need to be able to adapt. So suddenly the relative advantage of picking up random genetic change becomes more when there's an adverse environment. And so what we're doing is taking plant material, Sauvignon Blanc plant material, and basically distressing it, giving it a controlled stress treatment so that it removes that inherent silencing of its own mutation, and it becomes more genetically diverse just for a short period. We don't want these things to become to pick up more than one random change, because then you can end up with an improvement and a disadvantage at the same time. But as you say, you see that in the vineyard all the time. People will have seen plants that suddenly, in their gardens, a rose bush that usually does red roses, and suddenly there's a yellow one or something like that. It does happen, but that type of thing, like a color trait, is very easy to pick. We can pick Pinot Blanc from a Pinot Noir vineyard because it's easy to spot visually. But some of the traits we're looking for, like increased water use efficiency or productivity, that are quite subtle, even disease resistance. You just won't see it if there isn't a disease in that vineyard. So we need a way to be able to produce a collection of diversity that's more concentrated, where it's not 1 in 100. It's basically every plant is unique in that collection, and that we know this has been the diversity is represented in the smaller population. We can go and screen it for the traits that we want. But as you say, it's not directed, so we don't move in a certain direction towards one trait and then hope to see that at the end.
Yeah, presumably in the vineyard, you've got the vines sitting there in a high UV environment because, New Zealand has quite high levels of ultraviolet light. So presumably that's a little bit of mutagenesis that's happening out there. I mean, Emma, when you walk through the vines, do you ever notice vines that look, particularly different or unusual? Is it something that's, you've ever thought, oh, this is different, this one's performing better, maybe we should select, some cuttings from this and propagate those?
Yeah, look, Jamie, I think certainly from block to block, you might see a bit more of that variation. It's quite, it can be quite distinctive. And through walking through the rows, you do certainly see a bit of mutation. You might see a red bunch there, or something a bit strange. But I think what's really exciting about what the team at BRI are doing is they're creating these vine changes under such a rapid time frame that we've not been able to see before. And I think that's what's so cool about this innovative technique and management of it, is that we are really going to be able to push the boundaries of Sauvignon Blanc, and things we'll be looking for will be, for example, looser bunch structures, which might help with pest and disease resistance, just getting the airflow through the vines, through the bunches a wee bit more. Flavour, of course, is really fundamental. So I think that's what's exciting about what this research is doing, and I think that's why you see there's a number of wineries and wine companies that are backing it, and that's what we're here to explore. Yeah,
I mean, that bunch architecture must be quite an important one for Sauvignon, because botrytis is a big issue, isn't it? For at the end of the season, you don't want your slip skin or your botrytis to kind of come in. But using that as a specific example, Daryl, presumably when you're breeding like this, when you're doing these sort of studies where you're looking to get some natural variation or accelerate it, if you have to wait 3 years for to plant the vines and then see what the bunch of grapes is like, that slows things down considerably. Are there sort of like markers that you can use, genetic markers, so you can just make you can accelerate the variation, then you can look at that material without having to plant it in a vineyard and wait 3 years just to do that initial screen?
There are it's not a perfect system, but it does massively accelerate the selection process, and that's largely because the efficiencies of doing DNA sequencing have just become so much better. The cost calculations are so much more improved by things like even things like COVID, but effectively the technology of having rolled out the ability to sequence a lot of different variants. So that's basically what we can do. And the advantage is, as soon as a plant is really tiny, we can take a single leaf, one of the earliest leaves. We only need about a square centimeter of tissue to be able to extract all of the DNA for all of the genes that plant has. So based on that, for example, if you're looking at water use efficiency, we know about 20 different genes that grapevine has that are associated with changes in drought resistance in other plants. And the great thing about plants is they're almost all the same, have the same genes. So if you find a gene that controls a certain pathway in one plant, you can study that same gene. So we can prioritize the plants based on where they have these genetic changes. And Emma mentioned flavors and aromas. We know quite a lot about the biochemical pathways that make up each of those flavor and aroma compounds. So we can prioritize all of those genes that produce all of those enzymes as ones to screen for changes in. So that allows us to do an early selection of this random diversity at a very early stage.
Yeah. So presumably that's a great advantage, because otherwise because afterwards it's a numbers game. But it's like,
exactly.
If you can whittle down the numbers when you've got a little seedling, it's a lot easier than taking something to plant in a vineyard and then wait 3 years to see how it performs when it starts producing grapes.
That's true, and obviously that what happens when it produces grapes is what really trumps everything else. At the end of the day, that's what matters. So you do still go through to that. Certainly, you can also eliminate a lot of plants that are either just genetically identical to what you start with, or have changes that are nowhere near any genes, and so it greatly improves the efficiency.
And so, how do you increase the rates? Are you using mutagenesis? What sort of mutagenesis are you using to kind of accelerate these these naturally occurring mutations to the point that you're getting more of them?
Yeah, so you mentioned UV early on, and that was one of the things we looked at first. We looked at if we expose grapevine tissue to higher UV treatments, do we see a greater rate of genetic diversity, more mutagenesis within the plant? And we tried temperature shocks and salinity shocks, a whole lot of different stresses. And interestingly, what had the greatest effect was growing the grapevine tissue with microorganisms from different environments collected from New Zealand vineyards. And so in the lab, in controlled doses, co-cultivating these plants with these microorganisms, they're not pathogens or anything. Most of them are yeast that were collected from the vineyard, had a great effect. And that's kind of led us to optimizing treatments that remove the silencing of a certain part of the DNA. Within the DNA, there are these mobile elements, parts of people call them jumping genes or transposons, which are in large numbers, thousands of elements sitting there between the genes, and when they move around, they shuffle the DNA. And whereas something like UV usually causes a DNA strand break, and when that's repaired, you sometimes get a small change. So if the genetic code was a book, you would change one letter in that book. Whereas if you change, when these transposable elements move around, you're shuffling whole paragraphs, and so that's much more likely to change the story. So our research has looked at what causes this inherent ability for the plant to respond to stress by shuffling its genetics to be hyperstimulated, and so we use those treatments in a regime that does include temperature shocks and transferring them from various media so that they have different controlled growth environments to optimize the removal of the natural repression they have on that. So they normally silence these elements. The plant doesn't want to change genetically when it's growing really well, but it does have the ability to respond to stress by turning off that silencing.
I see. This is much more interesting than simple sort of the old-fashioned mutagenesis studies, where they're just trying to cause changes
it with
and
radiation
just hope for the best. Yeah. No, that's really exciting. Yeah. And so, what stage are you at now with this project?
Yeah, we've really been scaling up this year. So we've now produced about 4,000 vines. They're really small. They're just coming out of going into pots and coming through the nursery at the moment. We previously spent a lot of time optimizing the treatments, and now that we have a treatment that we know produces quite a lot of diversity, we're going through scaling it up. And at the same time, we are increasing the throughput at which we can do the DNA sequencing. So we have a liquid handling robot to automate the extraction of the DNA from this large number of plants. We've just installed a new DNA sequencer so we can run through large numbers of samples and rapidly prioritize them, as you say, for ones that look like they have potential.
And Emma, what stage are you going to be able to get presumably you're going to work as an industrial partner, with your vineyards, trying out some of these vines. At what sort of stage are you going to get your hands on some potentially exciting new variants of Sauvignon Blanc?
Well, exactly when that occurs, that'll be up to Daryl to advise. But yes, the exciting part of this project is that once there are my understanding is once there are vines available that we're looking at pursuing, they will be then planted in vineyards and managed through the grape growing and winemaking process. And so that brings in that commercial element that is so important, working, hand in hand with producers to really ensure that the clonal material that we're obtaining is things that will be viable in the marketplace. Because ultimately, at the end of the day, it's about continuing to deliver that Marlborough Sauvignon Blanc style that our consumers love and adore around the world. And for us as winemakers and growers, that's the number one priority.
And presumably then you have the fun task of assuming these vines do get to the, I should say, when these vines do get to the stage they're being put into the vineyard, then you've got the fun bit of kind of matching to site. Because you presumably if you've got 1,000 hectares of vines under your control, presumably you're in quite a lot of the different sub-regions. And so that sort of process of matching to site will be kind of key, presumably. Yeah.
So you've got sub-region, you've got soil type. There's so many variables that we understand a lot about now, but we can sort of test the strengths of this clonal material. And then, of course, winemakers do love a trial. So I'm sure there'll be many more trials in the winery just to push the boundaries even further.
Mm. And Daryl, just to pick up on one thing you said. So you said something about the co-culturing the vines with yeasts, microbes that might be found in the vineyard. That sounds really interesting. I mean, what's the mechanism by which these microbes are affecting the vine?
It is really interesting. And I don't think we can say we know enough about what the mechanism is, because it works even when the yeasts are dead. It's really hard to cultivate small amounts of tissue with live microorganisms that the microorganisms just overtake and you can't recover the plant material. So it seems to be that it's dosage related, and also there's something in the exposure of this of these grapevine cells to the microorganisms that causes them to turn off their silencing at a higher rate. Mm-hmm. It is an unusual situation for them. They're very exposed. They're growing in a completely sterile environment, and then you're just introducing this big hit from a microorganism. So we know what happens to the grapevine cells. We know that the DNA usually has methyl groups, chemical modifications that repress these transposable elements from moving. And we know that basically what happens is those methyl groups get removed. And so they start to become active again. And there are certain tissue types and conditions in the field that this is comparable with. But exactly what the mechanism is between the exposure to stress and the removal of the stress response is really unknown. But it's basically an interesting adaptive response we're piggybacking. Evolution is basically those two things you talked earlier about sex. That's rearranging genetic information that's already there into a new set of individuals, and that's what the siblings are. But the other part of evolution is producing new genetic information. The reason apples and bananas look different is because there's actually traits and genes that are present in one that aren't present in the other. So those have to come about from natural rates of mutation. And well, that's usually a very slow process, and the selection is usually natural. It's usually it takes some sort of event to make one survive over another. We can speed up the process, and then we can provide an artificial selection. Actually go in and pick the vines that have the loosest bunches, for example.
So getting slightly technical for a moment, there's another way in which grapevines in the field can adapt to the environment, which is epigenetic modifications. So where do those fit in? Because presumably once you take a cutting or you do tissue culture, you've got reprogramming of any epigenetic modifications that may have occurred. And there's been, and I think it's probably a field in terms of viticulture that's quite early on, and everyone's hyping it up a bit. But it does seem that there are some sort of epigenetic modifications in, say, for instance, vines that have been growing in the ground for 20, 30 years that will change the way that the vine performs.
Yeah, absolutely. And probably a couple of points on that. So that methylation of the DNA is the epigenetics. That's that second layer of which genes are expressed and which aren't. And that at least the most common current belief in the scientific consensus at the moment is that evolved basically as a way to repress viruses and these transposable elements, stop mutations from happening when you don't want them to happen, and was then kind of co-opted to alter gene expression in relation to different environments. And so you're right, we do get epigenetic changes, and that's actually a good thing. They're as useful as genetic changes because they produce trait changes. There is a resetting of some types of epigenetic changes, and so that's when we do our DNA sequencing, we're actually really fortunate that the type of sequencer we've brought in actually can analyze the epigenetic changes at the same time. It's a very new technology, and so we can use changes in the epigenetic expression of certain genes to select one over another. But as you say, you also over time seem to see an epigenetic adaptation of the plant to its environment, and so that's quite a long-term thing, and is likely associated with why much older vines perform quite differently to vines that have been in the ground 5 to 8 years.
So I think this is very exciting. The what you've been sharing, both of you, promises a lot for the future. You know, as Emma mentioned, you know, we've got a changing climate, we've got a chaotic climate, and it's very different to you know, the situation when Sauvignon Blanc first went into the ground. I think it was 75. Was it 73 were the first vines in Marlborough, and Sauvignon was not until 75. Things have changed quite a bit since then. But you know, with all this in view, this project, if it does succeed, then it's very exciting to think of kind of like these various traits, like you know, giving winemakers something a lot more to play with to keep Marlborough Sauvignon kind of in that pole position that it currently has in the global Sauvignon scene. So any thoughts about a timescale? Is that a bad question to ask?
Yeah, I think in terms of timescale, probably the most challenging thing for us is that we end up with a lot of plants, but they're all different. So you have one of each. So while you can identify how well that one performs in quite an objective way with a stress treatment or in a nursery, you then have to propagate it. And as Emma said, you have to try it on multiple different sites. And so I think the best way to approach that type of project is not to try and make one better plant. We're going to replace the UCD1 with UCD 1.2, and everyone swaps them all out. What I think would be better is if we, at least in the early stages, we move towards sites that have a need for lower water use, pick a selection of a dozen new clones that are all showing improvements in water use. We grow them, we bulk them up as a lot, we grow them in different bays, and then we do that early selection. It allows us to increase the rate at which we scale a bit better, but we don't just put all our eggs in one basket. And as a geneticist, that is quite exciting, that we wouldn't just have this one individual that if something changes, it all falls over. Instead, we have a collection of, this is more of a true mass select, a selection of plants that are designed for this environment based on how they were selected from a random group of diversity.
Great.
And I think diversity is the key word there. Sorry, Jamie. It's about adding diversity and continuing to add diversity and adding depth to what we're doing as an industry. And so I think it's about not changing what we're doing per se, but adding layers and depth and breadth. So that's the exciting thing. And it's evolution, it's innovation, and it's certainly something Kiwis, I think, do quite well. So it's exciting to see the results that will come from this.
And on that positive note, I think we've reached the end of our time. So I'd just like to thank you both, Emma and Daryl, for your really insightful contributions. Thank you both.
Thank you, Jamie.
It's been a pleasure. Thanks a lot.
It's been great.
Yeah, lovely. Thank you.